Cnidaria is a phylum containing over 10,000 species of animals found exclusively in aquatic environments. They are predominantly marine species. Their distinguishing feature is cnidocytes, specialized cells that they use mainly for capturing prey. Their bodies consist of mesoglea, a non-living jelly-like substance, sandwiched between two layers of epithelium that are mostly one cell thick. They have two basic body forms: swimming medusa and sessile polyps, both of which are radially symmetrical with mouths surrounded by tentacles that bear cnidocytes. Both forms have a single orifice and body cavity that are used for digestion and respiration. Many cnidarian species produce colonies that are single organisms composed of medusa-like or polyp-like zooids, or both. Cnidarians' activities are coordinated by a decentralized nerve net and simple receptors. Several free-swimming cubozoa and scyphozoa possess balanced sensing statocysts, and some have simple eyes. Not all cnidarians reproduce sexually, with many species having complex life cycles of asexual polyp stages and sexual medusa. Some, however, omit either the polyp or the medusa stage. Cnidarians were formerly grouped with sea ctenophores in the phylum Coelenterata, but increasing awareness of their differences caused them to be placed in separate phyla. Cnidarians are classified into four main groups: the almost wholly sessile anthozoa, swimming scyphozoa, cubozoa, and hydrozoa, a diverse group that includes all the freshwater cnidarians as well as many marine forms, and has both sessile members, such as hydra, and colonial swimmers, such as the Portuguese mano, or Staurozoa, have recently been recognized as a class in their own right rather than a subgroup of Scyphozoa, and there is debate about whether Mixozoa and Polypodiozoa are cnidarians or closer to bilaterians. Most cnidarians prey on organisms ranging in size from plankton to animals several times larger than themselves, but many obtain much of their nutrition from endosymbiotic algae, and a few are parasites. Many are preyed on by other animals including starfish, sea slugs, fish and turtles. Many scleractinian corals, which form the structural foundation for coral reefs, possess polyps that are filled with endosymbiotic algae, while reef-forming corals are almost entirely restricted to warm and shallow marine waters. Other cnidarians can be found at great depths, in polar regions, and in fresh water. Fossil cnidarians have been found in rocks formed about 580 million years ago, and other fossils show that corals may have been present shortly before 490 million years ago and diversified a few million years later. Fossils of cnidarians that do not build mineralized structures are very rare. Scientists currently think that cnidarians, sea ctenophores and bilaterians are more closely related to calcareous sponges than these are to other sponges, and that anthozoans are the evolutionary aunts, or sisters, of other cnidarians, and the most closely related to bilaterians. Distinguishing features: cnidarians form an animal phylum that are more complex than sponges, about as complex as sea ctenophores, and less complex than bilaterians, which include almost all other animals. However, both cnidarians and sea ctenophores are more complex than sponges as they have cells bound by intercell connections and carpet-like basement membranes, muscles, nervous systems, and some have sensory organs. Cnidarians are distinguished from all other animals by having cnidocytes that fire like harpoons and are used mainly to capture prey. In some species, cnidocytes can also be used as anchors. Like sponges and sea ctenophores, cnidarians have two main layers of cells that sandwich a middle layer of jelly-like material, which is called the mesoglea in cnidarians. More complex animals have three main cell layers and no intermediate jelly-like layer. Hence, cnidarians and sea ctenophores have traditionally been labeled diploblastic, along with sponges. However, both cnidarians and ctenophores have a type of muscle that, in more complex animals, arises from the middle cell layer. As a result, some recent textbooks classify ctenophores as triploblastic, and it has been suggested that cnidarians evolved from triploblastic ancestors. Description: basic body forms: abral end, oral end, mouth, oral end, abral end, ectoderm, gastroderm, mesogly, digestive cavity. Adult cnidarians appear as either swimming medusa or sessile polyps, and many hydrozoan species are known to alternate between the two forms. Both are radially symmetrical, like a wheel and a tube respectively. Since these animals have no heads, their ends are described as oral and abral. Most have fringes of tentacles equipped with cnidocytes around their edges, and medusa generally have an inner ring of tentacles around the mouth. Some hydroids may consist of colonies of zooids that serve different purposes, such as defense, reproduction and catching prey. The mesogly of polyps is usually thin and often soft, but that of medusa is usually thick and springy, so that it returns to its original shape after muscles around the edge have contracted to squeeze water out, enabling medusa to swim by a sort of deck propulsion. Skeletons in medusa: the only supporting structure is the mesogly. Hydra and most sea anemones close their mouths when they are not feeding, and the water in the digestive cavity then acts as a hydrostatic skeleton, rather like a water-filled balloon. Other polyps, such as Tubularia, use columns of water-filled cells for support. Sea pens stiffen the mesogly with calcium carbonate, spicules and tough fibrous proteins, rather like sponges. In some colonial polyps, a chitinous periderm gives support and some protection to the connecting sections and to the lower parts of individual polyps. Stony corals secrete massive calcium carbonate exoskeletons. A few polyps collect materials such as sand grains and shell fragments, which they attach to their outsides. Some colonial sea anemones stiffen the mesogly with sediment particles. Main cell layers: Cnidaria are diploblastic animals. In other words, they have two main cell layers, while more complex animals are triploblasts, having three main layers. The two main cell layers of cnidarians form epithelia that are mostly one cell thick, and are attached to a fibrous basement membrane, which they secrete. They also secrete the jelly-like mesogly that separates the layers. The layer that faces outwards, known as the ectoderm, generally contains the following types of cells: epitheliomuscular cells whose bodies form part of the epithelium but whose bases extend to form muscle fibers in parallel rows. The fibers of the outward-facing cell layer generally run at right angles to the fibers of the inward-facing one. In Anthozoa and Cyphozoa, the mesogly also contains some muscle cells. Cnidocytes, the harpoon-like nettle cells that give the phylum Cnidaria its name. These appear between or sometimes on top of the muscle cells. Nerve cells. Sensory cells appear between or sometimes on top of the muscle cells, and communicate via synapses with motor nerve cells, which lie mostly between the bases of the muscle cells. Interstitial cells, which are unspecialized and can replace lost or damaged cells by transforming into the appropriate types. These are found between the bases of muscle cells. In addition to epitheliomuscular, nerve and interstitial cells, the inward-facing gastroderm contains gland cells that secrete digestive enzymes. In some species it also contains low concentrations of cnidocytes, which are used to subdue prey that is still struggling. The mesogly contains small numbers of amoeba-like cells, and muscle cells in some species. However, the number of middle layer cells and types are much lower than in sponges. Polymorphism refers to the occurrence of structurally and functionally more than two different types of individuals within the same organism. In hydrozoans, colonial individuals arising from individual zooids will take on separate tasks. For example, in abelia there are feeding individuals, the gastrozoids, the individuals capable of asexual reproduction only, the gonozoids, blastostyles, and free-living or sexually reproducing individuals, the medusa. Cnidocytes. These nettle cells function as harpoons, since their payloads remain connected to the bodies of the cells by threads. 3 types of cnidocytes are known. Nematocysts inject venom into prey, and usually have barbs to keep them embedded in the victims. Most species have nematocysts. Spirocysts do not penetrate the victim or inject venom, but entangle it by means of small sticky hairs on the thread. Tichocysts are not used for prey capture. Instead, the threads of discharged tichocysts are used for building protective tubes in which their owners live. Tichocysts are found only in the order Cereanthria, tube anemones. The main components of a cnidocyte are a cilium which projects above the surface and acts as a trigger. Spirocysts do not have cilia. A tough capsule, the cnida, which houses the thread, its payload, and a mixture of chemicals that may include venom or adhesives or both. A tube-like extension of the wall of the cnida that points into the cnida, like the finger of a rubber glove pushed inwards. When a cnidocyte fires, the finger pops out. If the cell is a venomous nematocyte, the finger's tip reveals a set of barbs that anchor it in the prey. The thread, which is an extension of the finger, uncoils round it until the cnidocyte fires. The thread is usually hollow and delivers chemicals from the cnida to the target. An operculum over the end of the cnida. The lid may be a single hinged flap or 3 flaps arranged like slices of pie. The cell body, which produces all the other parts. It is difficult to study the firing mechanisms of cnidocytes as these structures are small but very complex. At least 4 hypotheses have been proposed. Rapid contraction of fibers round the cnida may increase its internal pressure. The thread may be like a coiled spring that extends rapidly when released. In the case of chironeck, chemical changes in the cnida's contents may cause them to expand rapidly by polymerization. Chemical changes in the liquid in the cnida make it a much more concentrated solution, so that osmotic pressure forces water in very rapidly to dilute it. This mechanism has been observed in nematocysts of the class Hydraza, sometimes producing pressures as high as 140 atmospheres, similar to that of scuba tanks, and fully extending the thread in as little as 2 milliseconds. Cnidocytes can only fire once, and about 25% of a hydra's nematocysts are lost from its tentacles when capturing a brine shrimp. Used cnidocytes have to be replaced, which takes about 48 hours. To minimize wasteful firing, 2 types of stimulus are generally required to trigger cnidocytes. Nearby sensory cells detect chemicals in the water, and their cilia respond to contact. This combination prevents them from firing at distant or non-living objects. Groups of cnidocytes are usually connected by nerves, and if one fires, the rest of the group requires a weaker minimum stimulus than the cells that fire first. Locomotion. Medusa swim by a form of jet propulsion. Muscles, especially inside the rim of the bell, squeeze water out of the cavity inside the bell, and the springiness of the mesogle powers the recovery stroke. Since the tissue layers are very thin, they provide too little power to swim against currents and just enough to control movement within currents. Hydras and some sea anemones can move slowly over rocks and sea or stream beds by various means, creeping like snails, crawling like inchworms, or by somersaulting. A few can swim clumsily by waggling their bases. Nervous system and Census. Cnidaria have no brains or even central nervous systems. Instead, they have decentralized nerve nets consisting of sensory neurons that generate signals in response to various types of stimulus, such as odors, motor neurons that tell muscles to contract, and cobwebs of intermediate neurons to connect them, as well as forming the signal cables. Intermediate neurons also form ganglia that act as local coordination centers. The cilia of the cnidocytes detect physical contact. Nerves inform cnidocytes when odors from prey or attackers are detected and when neighboring cnidocytes fire. Most of the communications between nerve cells are via chemical synapses, small gaps across which chemicals flow. As this process is too slow to ensure that the muscles round the rim of the medusa's bell contract simultaneously in swimming, the neurons which control this communicate by much faster electrical signals across gap junctions. Medusa in complex swimming colonies such as siphonophores and chondrophores sense tilt and acceleration by means of statocysts, chambers lined with hairs which detect the movements of internal mineral grains called statoliths. If the body tilts in the wrong direction, the animal rights itself by increasing the strength of the swimming movements on the side that is too low. Most species have ocelli, which can detect sources of light. However, the agile box jellyfish are unique among medusa because they possess 4 kinds of true eyes that have retinas, corneas and lenses, although the eyes probably do not form images. Cubozoa can clearly distinguish the direction from which light is coming as well as negotiate around solid-colored objects. Feeding and excretion. Cnidarians feed in several ways. Predation, absorbing dissolved organic chemicals, filtering food particles out of the water, and obtaining nutrients from symbiotic algae within their cells. Most obtain the majority of their food from predation, but some, including the corals Heteroxenia and Leptogorgia, depend almost completely on their endosymbionts and on absorbing dissolved nutrients. Cnidaria give their symbiotic algae carbon dioxide, some nutrients, a place in the sun and protection against predators. Predatory species use their cnidocytes to poison or entangle prey, and those with venomous nematocysts may start digestion by injecting digestive enzymes. The smell of fluids from wounded prey makes the tentacles fold inwards and wipe the prey off into the mouth. In medusa, the tentacles round the edge of the bell are often short and most of the prey capture is done by oral arms, which are extensions of the edge of the mouth and are often frilled and sometimes branched to increase their surface area. Medusa often trap prey or suspended food particles by swimming upwards, spreading their tentacles and oral arms and then sinking. In species for which suspended food particles are important, the tentacles and oral arms often have rows of cilia whose beating creates currents that flow towards the mouth, and some produce nets of mucus to trap particles. Their digestion is both intra- and extracellular. Once the food is in the digestive cavity, gland cells in the gastroderm release enzymes that reduce the prey to slurry, usually within a few hours. This circulates through the digestive cavity and, in colonial cnidarians, through the connecting tunnels, so that gastroderm cells can absorb the nutrients. Absorption may take a few hours, and digestion within the cells may take a few days. The circulation of nutrients is driven by water currents produced by cilia in the gastroderm or by muscular movements or both, so that nutrients reach all parts of the digestive cavity. Nutrients reach the outer cell layer by diffusion or, for animals or zooids such as medusa which have thick mesoglea, are transported by mobile cells in the mesoglea. Indigestible remains of prey are expelled through the mouth. The main waste product of cells' internal processes is ammonia, which is removed by the external and internal water currents. Respiration. There are no respiratory organs, and both cell layers absorb oxygen from and expel carbon dioxide into the surrounding water. When the water in the digestive cavity becomes stale, it must be replaced, and nutrients that have not been absorbed will be expelled with it. Some anthozoa have ciliated grooves on their tentacles, allowing them to pump water out of and into the digestive cavity without opening the mouth. This improves respiration after feeding and allows these animals, which use the cavity as a hydrostatic skeleton, to control the water pressure in the cavity without expelling undigested food. Cnidaria that carry photosynthetic symbionts may have the opposite problem, an excess of oxygen, which may prove toxic. The animals produce large quantities of antioxidants to neutralize the excess oxygen. Regeneration. All cnidarians can regenerate, allowing them to recover from injury and to reproduce asexually. Medusa have limited ability to regenerate, but polyps can do so from small pieces or even collections of separated cells. This enables corals to recover even after apparently being destroyed by predators.